My name is James Schnable. I'm an associate professor here at UNL. I've been at the university a little over 6 years now. The work in my lab focuses on maize and sorghum, and what really sets us apart is that we are what I would describe as a full stack lab. So we have people doing everything from very conventional molecular biology, genomics and bioinformatics, conducting high-throughput phenotyping in the greenhouse here on campus and out in the field, as well as people conducting yield trials across the state of Nebraska. So I've been working with Psomagen since before I started at UNL back when it was Macrogen USA, I think.
My name is Guangchao Sun. I'm a postdoc in Dr. Schnable Lab. I have been working here for 3 years. My research has been focused on understanding the genetic control of maize adaptation to environments using comparative genomics and quantitative genetics and some molecular biology approaches.
My name is Jonathan Turkis. I've been working in James Schnable's lab for the past 8 months. My role in the lab is a research technician, managing a lot of the molecular biology work that's going on in the lab.
One of the projects in my lab is looking at both natural variation in corn and in sorghum for nitrogen use efficiency, and looking at how does the plant change its metabolism, how does it compensate, I don't have enough of this resource, what can I do instead, or how can I be more efficient? And then figuring out how we could move these compensation strategies that have evolved or been selected for in low-yielding crop varieties or crop wild relatives into the high-performing varieties that we grow here in Nebraska today.
The plants cannot move, so they need to deal with different kind of environmental stresses like drought, like cold, or nutrients deficiency, or this kind of thing. So I wanted to understand like how plants deal with that. I want to understand the molecular mechanisms underlying these responses. So when James mentioned this company, when I had a plate of a pre-made low-pass sequencing library for sequencing, that was for 96 sorghum individual. And the low-pass DNA sequencing is very good technology that we could generate a genotype information for the whole genome for the whole population at a relatively low cost. The first time when I interacted with Psomagen, that was kind of a challenge to me because that might be my first experience of preparing, first of all, low-pass DNA sequencing library.
In my experience so far, I have not prepared libraries on my own. We've always hired Psomagen to do those services for us. It ends up being more cost effective because while potentially some money can be saved by us doing it, there's a lot of time that is involved with it. And by providing it with Psomagen, people who do this stuff every day, they're much better at it. The projects that I've been working with in James's lab have been my first experience with being involved with sequencing at all. And I've had a lot of questions. I've been able to finally grasp a lot of the different concepts, the different machinery that's involved, QC, all that kind of thing. Things that would have taken me so much longer to learn if I had to do it on my own.
I asked a lot of questions and I always got very positive feedbacks and also the detailed instructions.
That's why I really like working with competent service providers. It saves my people time, which allows the lab to keep running on the budget we have and make more progress. It's also a lot more fun for the people actually doing the work.
We are aware of the progress of the whole process, and that's very important for us to know because our samples might be temperature sensitive, might be time sensitive. If the samples are not properly handled, we're gonna get worried and concerned.
We really value our relationship with Psomagen, particularly the fact that the turnaround time for sequencing is so low. Often we're waiting on data that we need to then analyze to make decisions about what new varieties are going to go into the field in the next year or what new experiments are going to happen.
Psomagen will send us a summary of essentially everything that was done. That makes it so much easier because it makes allowing you to write a methods section of a paper much easier.
So whenever we can take something that isn't fundamental to the actual design of the experiment and work with somebody who's a real expert in that area instead of trying to muddle through ourselves, I think it's good for the students, it's good for the budget of the lab, and it's good for the people depending on the output of that research.
Psomagen is the first choice as the only option that we considered. So we trust you, and that's why we choose you.